Um, Today I would like to, well, first of all, thank you. I would like to thank the organizers for giving me this opportunity. Today I will try to convince you that individuality is ubiquitous at all levels in biology, and also drive you home with the fact that fat, rubber, and logo have something in common. Well, individuality as humans is obvious, coming from our different DNA and our different environment. And this individuality coming from our DNA is becoming more obvious and more understandable thanks to the Human Genome Project. So, but twins, for instance, have the same DNA, so they share the same nature. So it's an interesting model organism to decouple the effects of the environment from the effects of the genetic background. So that's why in psychology there has been a lot of twin studies to understand the effect of environment on twins that are separated from each other very early on in the childhood. Okay, well, humans are very complex to understand. Do we have a simpler system? Well, we have been looking at this very tiny molecule, some RNA molecule from a virus. And we have been attaching probes on it to understand the movements of this RNA molecule. What strikes us as something interesting is that, well, and bear with me that these traces resemble the behavior of the molecules. And as you can see, the nature of the traces are very different. So seemingly identical molecules within the same environment display different behavior. Well, in a lighthearted analogy, this is like penguins, that they seemingly identical penguins may behave very differently. Maybe you find one that's flapping its wings all the time, whereas the other remains silent most of the time. So when we presented this data, for instance, people really asked the very same question. How do you know that this is not coming from the fact that you are not affecting the molecules when looking at them? So all in all, I tried to come up with an alternative strategy to look at the molecules for a long time using some fats. So in analogy, again, to the penguins, the penguin is now, instead of being tethered to a surface, is now confined in a bubble, which can move freely in the bubble. So its behavior is not affected. So we saw that the molecule-to-molecule variation is real. Okay, but why is it there? And does it do anything useful? The long answer, to give a long answer to that, we have some speculations, but the short answer is we don't know. But I will still try to keep my promise and convince you that this variability is there, but also tell you that it does something, sometimes does something useful. But all in all, well, the variability comes from nature or nurture or chance, as shown by these RNA molecules. So now I would like to switch gears to a different system, which we understand better. So in my postdoctoral studies, I started working on the cell-to-cell variation. And we worked on some bacteria, which is really a typical model organism in biology, E. coli. Okay, so the dream of a bacterium is to become two bacteria. And this was stated by Francois Jacob, the Nobel Prize laureate in 1965. So essentially that means when you have one bacteria, well, simply because a bird is cuter to look at. And if there is like enough food around, the bacteria will continue to divide as long as the food is there. But then rarely something happens to one bacteria that it goes into an hibernating state, that it actually stops dividing. And this, again, I cannot emphasize this enough, that all these bacteria share this very same gene. So they have identical DNA sequences, and they reside in identical environments. But all of a sudden, one becomes different. So everybody keeps dividing, there is food around, everybody's happy, but this guy. So I can see from your faces that you feel the pity for this guy because, well, there is, everything is good for everyone, everybody is enjoying a big feast. But this guy, and you should feel sorry, but every cloud has a silver lining. So when actually an antibiotic treatment appears, or some kind of stress actually hits upon the bacterial population, this is a catastrophe, and the only survivor is our guy. So basically, this very bacteria, by just going into that hibernation mode, evades the antibiotic treatment. And then, as I told you, this is just a transient state. So our bacteria, our guy now wakes up, or if it happens to wake up, when the antibiotics is washed out, it can regenerate the population. And then maybe at some time, it's some other bacteria's turn to go into that hibernation mode. So if you will, the bacteria actually go into this hibernation mode, and they take turns to make the sacrifice. What's that? So this is actually, I will come to that. It's different from the resistance. So the rarely occurring non-dividing bacterium persists in the case of a threat and ends up regenerating the whole population. This may not be good for the individual bacterium, but good for the population. This is the insurance policy of the bacterial population, if you will. They reserve a tiny fraction of the individuals to sleep and then take turns in doing so. And the switching between non-dividing and dividing states happens by chance. And note that the hibernating fraction must be very low because the population wants to grow as fast as possible. So basically, we are trying to understand what derives the switch and how frequently it happens and all that. And I will not go into very technical details. But then, okay, we were talking about the optimists and the pessimists. And the optimists say every cloud has a silver lining. So that was a good, a story with a good ending for the bacteria. But the pessimists say every silver lining has a cloud. So in retrospect, what is good for the bacteria's point of view is bad for us, or it is bad for the patients, because this is a common problem in antibiotic treatment, and actually even in cancer therapies. That's fractional killing. So this is why people are encouraged to use the antibiotics as prescribed. So the idea is to hit them hard and continuously until you wipe off everything. Wipe off the whole infection, because anything that evades the infection will bring it back. So this just explains your question, that the difference between resistance and these persisters. In the case of resistance, the bacteria actually becomes something else. There is a permanent mutation. And when they cross what, as Dr. Kenenstein describes, the valley of death, beyond the valley of death, now you have the population that is different from the starting population. So this is the antibiotic resistance. And what is so-called as, as the well-known name goes, superbugs in the hospitals. This is something different. This is just a temporary change. And these are called actually persisters in the technical term. And the population actually is not different from the initial population after the antibiotic threat. So what we do is basically to understand these things better, we do plumbing at microscale. We build chips with tiny flow channels, so-called microfluidics. And for that, we use some rubber. And we're essentially looking for the needles in the haystack, these rare cells in the population. And these are all single bacteria. And then, last but not the least, Lego Mindstorms come to the rescue for screening. So we basically generated this super cheapo liquid handler in an automated fashion to be able to like screen these very widely used 96-well plates. So I hope I convinced you that fat, rubber, and Lego can all be used to study individuality in biology. And thank you.